Yes, good morning everyone. Good morning, whether I'm audible? Whether I'm audible? Yes. Today we are going to discuss PYQ for year 2020. Okay? This is the last question paper. Today we are going to discuss. Okay? Be attentive in class. If you have any doubt, kindly ask me. Okay? First question what they are asking. Which type of RNA molecules makes up more than 80% of total RNA in the cells? More than 80% of total RNA in the cell is made up of which type of RNA? Yes, ribosomal RNA. Very good. Okay. Ribosomal RNA will be present approximately 80% of total RNA in the cells. Okay. Then next most abundant is tRNA. Okay. tRNA will be around approximately 15 to 20% will be there. Okay. Then the most least content is mRNA. It will be there around 5 to 10%. Okay. Then another important thing we have to discuss here is the smallest RNA molecule is smallest RNA molecule among ribosomal RNA, mRNA and tRNA is tRNA. Okay. tRNA is the smallest among mRNA, tRNA and ribosomal RNA. This is also another important point you have to remember. Okay. What is the bond angle? The HOH bond angle in water is what? What will be the answer? The HOH bond angle in water is 103 degree, 104.5, 106. Yes, very good. 104.5 degree will be there. This we have all discussed in the class. Okay. Then another important point, water molecule will be joined with another water molecule by hydrogen bond. Okay. This is another important point. Hydrogen bond will be there between the water molecule. Okay. In water molecule, the hydrogen and the oxygen will be joined through which bond? Covalent bond. Okay, covalent bond. This is also another important point you have to remember. This bond angle is 104.5 degree. This what they have asked the question. Okay. The chromosome and complement of 2n minus 2 is what? General agree question. 2n minus 2. Chromosome and complement of 2n minus 2. Whether it's monosomy, trisomy, nullisomy or double nullisomy. What will be the answer? Yes, very good, very good. Nullisomy. Okay. So, monosomy means what? 2n minus 1 is monosomy. Yes. Trisomy means what? 2n plus 1. Nullisomy means 2n minus 2. In our question, they have asked complement of 2n minus 2. So, nullisomy will be the right answer. Then what is meant by double nullisomy? 2n minus 2 and minus 2. This is double nullisomy. Yes, very good students. Next, the growth rate which is also called as efficiency index. Okay, they have given different growth parameters, CGR, NAR, AGR and RGR. Among these parameters, which we will call as efficiency index, index, whether RGR or AGR, yes, very good. We can call relative growth rate as efficiency index. Okay, relative growth rate as efficiency index. What is meant by relative growth rate? It's a growth increase relative to the existing plant weight. Okay, growth increase relative to the existing plant weight is known as RGR. And the unit for RGR, this is also another important thing. Unit for RGR is gram per day. Okay, this is also another important point you have to remember. Okay, from growth parameters, sure they will ask one question. Okay, next, which of the following is an omega-9 fatty acid? They are asking about omega-9 fatty acid. What will be the right answer? Omega-9 fatty acid. This we have already discussed. If you see erythric acid, 22 carbon will be there, one double bond will be there at position 13th. Palmitoleic acid means what? 16 carbon will be there and double bond will be present at position number 1. Okay. Gamma linolenic acid, 18:3. Del 6, 9 and 12. Eicosapentaoic acid means what? EPA. This is also important point. I am writing here now. This is also important point. C20 double bond will be 4 will be there. Sorry, 5 double bond will be there. Okay. So if you see among this, which is omega 9 fatty acid. So omega 9 fatty acid is what? Yes, very good. Erucic acid is omega 9 fatty acid. So palmitoic acid is what? It's an omega 7 fatty acid. Okay. Gamma linoleic acid is what? It's omega 8 fatty acid. Eicosapentaenoic acid is what? Omega 3 fatty acid. This also you have to remember. Okay, clear? Next, the enzyme involved in removal of RNA primers in DNA replication process. Which enzyme is important for removing RNA primers during replication process? Yes, whether it's a DNA polymerase or DNA primase or endonuclease or RNA polymerase. Yes, DNA polymerase. We have already seen DNA polymerase 1, particularly if you see DNA polymerase 1, this will have three different, yes, it will have three different activities will be there. 5' to 3' polymerase activity, 5' to 3' exonuclease activity, then 3' to 5' polymerase activity. So this primers will be removed by 5' to 3' exonuclease activity. So the right option is DNA polymerase. Which of the following cannot be considered as climacitic fruit? This is another repeated question. Which is not the example of climacitic fruit among these? Citrus, banana, mango and apple. Yes, very good. Citrus is the typical example for non-climacitic fruit. Apart from that, we can say grapes, okay, orange. These are some of the example of non-climacitic fruit. Yes, these are some of the example of non-climacitic fruits. Another question from general agri. Okay. Chromosomes are relaxed to the maximum at which stage of cell cycle? Whether interphase, telophase, metaphase or prophase. Which state? Under which stage? Yes, very good. At the interphase stage, if you see here, interphase, prophase, metaphase, anaphase and telophase. In interphase, the chromosome will be what? It will be highly relaxed state will be there. Okay. Why it is there in highly relaxed state? Means for promoting the DNA replication process. Okay. So in interphase, it will be present in relaxed form. Okay. Relaxed form, it will be present. Okay, students. Next. Another important question from enzymology, assertion and reason. Sorry, statement question they have asked. The catalytic efficiency of an enzyme cannot exceed the diffusion control rate. Catalytic efficiency of an enzyme cannot exceed the diffusion control rate of combination of enzymes and substrate to form enzyme substrate complex. You have to observe the question. They are saying if enzyme is binding with substrate, this will lead to formation of enzyme substrate complex. This will be breakdown to enzyme and product. Okay. They are saying this catalysis will not exceed. This will not exceed the diffusion control rate. Yes, this is true only. This we have already seen in our enzymology class. The first statement is true only. The catalytic efficiency of enzyme is defined as ratio of kcat by km. Yes, this is also true. So both statement 1 and 2 are true. Yes, very good. The energy bond in double helical structure of DNA, which are responsible for holding the two strands together or what? They are saying the two strand of DNA is there. What is the energy that is holding these two strands together? What will be the energy? Yes, very good. Hydrogen bond. Hydrogen bond will be there. That will be hold this two strand of the DNA together. Yes. Hydrogen bond between the bases. A will always pass with T. G will pass with C with two hydrogen bonds and with three hydrogen bonds. Okay. Then most important thing you have to remember here. Another thing, the nucleotides. Nucleotides will be joined together by which bond? Phosphodiester bond. Okay. The nucleotides will be joined by phosphodiester bonds. Okay. Which of the following element can cause necrosis of young meristematic region? They are saying necrosis of young meristematic region when present in insufficient amount for plant physiological process. What will be the element? Yes, very good. Calcium. Calcium is typical element. If the deficiency is there, means that will cause necrosis at the young meristematic tissue. Why it is happening at young meristematic tissue? Because calcium is immobile in plant. Okay, because of that, symptoms are happening at young meristematic regions. Yes, very good. Which of the following nucleosides have been shown to functioning as a local hormone to control various physiological functions in human? Signaling class, we have already discussed this one. Which of the following nucleoside? Whether it is an adenosine or inosine, guanosine or cytosine? Yes, very good. Adenosine. It will act as a signaling molecule, a local signaling hormone that will enhance the synthesis of other hormones for physiological functioning in mammals. Yes, clear. Doubled haploid is a plant. Okay, a double haploid plant is will be whether it's an heterozygous or homozygous or always vigorous or always true breeding in cross-pollinated species. What will be the answer? Doubled haploids. Yes, doubled haploids will always be homozygous in nature. Okay, it will be always homozygous. Okay, this is another important point that anther culture we will use. Okay, this thing they will use for production of double haploid. They will treat the compounds, they will treat with colchicine treatment, because of that chromosome doubling will be happened. Okay, clear. In vegetative tissue and seeds, phosphorus is stored in which form? In which form phosphorus will be stored? Whether it will be stored as gluten, phytin, calmodulin or zyme. Yes, gluten, phytin, yes, very good, phytin. Okay. If you see the structure of phytin, this phytin, we can also say as phytic acid. Okay, phytic acid. This we can also say as myoinositol, hexakis phosphate. Okay, hexakis phosphate. Okay, this is another important point you have to remember. Okay, why means there will be a myoinositol structure will be there. Here 6 molecule of phosphorus will be attached in this structure. Okay, the most important thing about this myoinositol phosphate is, okay, this will have excessive negative charges. Okay, so this negative charge will chelates the positive charge ions like calcium and magnesium. Then it will decrease the bioavailability of these nutrients in body. So this may act as an anti-nutritional factor. Okay, so phytic acid is an anti-nutritional factor, but for plants it will act as a storage form of phosphorus. Clear? This is very important point. Okay, which of the following sugar enter in the glycolysis via Leo layer pathway? Okay, feeder pathway, Leo layer pathway. Okay, which of the following sugar is entering into glycolysis via Leo layer pathway? What will be the answer? Whether fructose, mannose? Yes, very good. Galactose. Okay, galactose will be enter into glycolysis by Leo layer pathway. This is very important point. Okay, first the galactose, by galactokinase it will convert to galactose 1 phosphate. Then this galactose 1 phosphate will react with UDP glucose. Okay, this is very important point. By this process, this glucose 1 phosphate will be released. This will be targeted to glycolysis pathway. Okay, so Leo layer pathway is primary pathway that will involve for transferring the galactose to glycolysis pathway. Okay. Next, the most active metabolite of vitamin D, that means biological active form of vitamin D, which helps in regulation of calcium and phosphorus metabolism is what? Biologically active form of vitamin D that is regulating calcium and phosphorus metabolism. Whether it's 1-hydroxycholesterol, 25-hydroxycholesterol or 1,25-hydroxycholesterol. Yes, very good, very good. 1,25-dihydroxy cholecalciferol. This we can also say as calcitriol. Why we are saying calcitriol? Because of presence of 3-hydroxy group. At which position? 125 and 3. Okay. That's why we are saying calcitriol. This is the biologically active form of vitamin D that will regulate the calcium and phosphorus metabolism. Clear? The term biological value, we can also say its economical yield, was given by whom? Whom has given this term biological value? Yes, general agri question. Yes. Yes, very good, very good. Yes. Biological value, the term was given by Nitsporvachi. TK, he is the person given. This we can also say as economical yield. This formula also they have, this formula also we have already discussed in our class. Economical yield by biological yield. Okay, biological yield. That means it is harvest index. Okay. So the term was given by Nitsporvachi. Okay. Sir, when you will take the classes for SRF Biotechnology, I will let you know. Okay, Safura Javed. Sure, we will let you know. Okay. The first step of Calvin cycle in C4 plant is what? Whether carboxylation will happen or regeneration or protonation or reduction. What it will happen? Carboxylation. Yes, very good. First step is carboxylation. We know carbon dioxide will be there. Okay. That will be converted to bicarbonate first. Okay. This will enter into mesophile cells. Okay, mesophile cells will be there. First, what enzyme will come? Pep carboxylase will come. Okay, pep carboxylase will enzyme will come. This will convert this PEP to oxaloacetate. So first step is what? Carboxylation is the first step. Okay, yes, very good. Which of the following plant does not belong to family of camp plants? What will be the answer? Among the plants, which is not a camp plant? Which is not a camp plant, whether pineapple, agave, sugarcane or cacti. Yes, very good, sugarcane. Because sugarcane is a C4 plant. Sugarcane, maize are C4 plants. Yes, sugarcane, maize are C4. Which of the following, this they have asked in 2021 also, we have seen yesterday only. Which of the following vitamin is required for conversion of deoxyuridine diphosphate to deoxythymidine diphosphate? What will be the answer? Which element, which vitamin is required? Yes, very good. For conversion of deoxy-UDP to deoxy-TDP, we need folate. Why folate is required here? Apart from that riboflavin, flavin and thymine. Why folic acid? We know this folic acid is primarily involved in single carbon transfer reactions. Okay? Single carbon transfer reaction, particularly in reduced form. Okay? Particularly in reduced form. Okay? If you see the conversion of DUDP to deoxy-TDP, tetrahydrofolate will be there. Okay? Tetrahydrofolate will be there. Okay? This will donate methyl group CH3 group to carbon 5 of uracil molecule. Okay. We know the basic difference between uridine and thymine is what? At fifth position we have extra CH3 group. This will be donated by this tetrahydrofolate. That's why folic acid is involved in there. Okay, clear? Then biotin means what? This will be primarily involved in carboxylation reactions. Yes, riboflavin means what? That is vitamin B2. Okay, for oxidation reduction reaction, it will be involved. Thiamine pyrophosphate, TPP. I told yesterday itself. This will be primarily involved in aldehyde transfer reactions. Aldehyde transfer reaction, particularly if you see transaldolase, transketolase. This enzyme will have thiamine pyrophosphate. That means TPP as a cofactor. Okay, clear? In photosystem 1, the pigment molecule absorb maximum light at which nanometer? Whether it will absorb maximum at which nanometer? 660, 680, 700 and 720. Yes, very good. 700. PS1 is 700. PS2 is 680. Okay, in some of the book they have mentioned 680 as well as 690 nanometer also. Okay. Then you have to read about Heliobacter. What will be the absorption for Heliobacter? Then bacteriochlorophylls. Bacteriochlorophylls, what are the absorption? This also you have to read once. Okay, just you go through once. Okay, students. Okay. Tumor cells can identified in PET. PET means what? Positron emission tomography scans. Okay. A site of usually high amount of 18 fluorine labeled fluoro 2 deoxyglucans accumulation is there. Why? Why it is there? Okay. Observe the question they are asking about glycolysis. Okay. In cancer cells, there will be a more utilization of glucose as a energy source because this cancer cells will always grow under anaerobic condition. Okay. If anaerobic condition is there means pyruvate will not be oxidized to acetyl CoA and further to carbon dioxide and water. So the cancer cell will particularly depends on glycolysis as sole source of energy. Okay. That will particularly depend on glycolysis as source of energy. So there will be higher consumption of glucose molecule in cancer cells. Okay. If we provide this compound that is fluorine labeled 2-fluorodeoxyglucose, that will be accumulated. Okay, that may be uptaken by the glycolysis enzyme, but accumulated at particular tissues. Wherever the cancer tissues are there, there it will accumulate. Okay, by this we can know wherever the cancer tissues are present in the body. Okay, so why it is happening means because of the high rate of glycolysis. Okay, because of high rate of glycolysis. This is another important question in cancer cells. Always it will be anaerobic. Okay, clear? How many protons are released on cytoplasmic side of inner mitochondrial membrane by complex 3 for every 2 protons taken up from matrix side? They are saying how many protons will be given or how many protons will be released. Yes, we know complex 1, 2, 3 and 4 will be arranged in like this. Okay. I already told in our electron transport chain class itself. Complex 1 will pump 4H plus ions, complex 3 will pump 4H plus ions, and complex 4 will pump 4H plus ions. Yes. So complex 3 will pump 4H plus ions to cytosol side of inner mitochondrial membrane. Yes, very good. Okay, 4H plus. Okay. An enzyme involved in initiation of DNA synthesis during replication. Which enzyme will be involved for initiating the DNA synthesis during replication? Whether it's telomerase, holoenzyme, or RNA primase or endonuclease. What will be the answer? Yes, very good. Very good. RNA primase. Okay, we know for DNA replication, RNA primers are required to initiate the synthesis. Okay. RNA primers are required to initiate the synthesis. These primers are synthesized by primase enzyme, RNA primase enzyme. It is removed by, that primers will be removed by DNA polymerase 1 enzyme. This question also they have already asked in same year only. We have already discussed that question. Okay, clear? Scientists who have given the name mitochondria, who has given the name for mitochondria? Whether Richard Altman, Carl Benda, Andrew Schimper or George Pallet, who has given? Scientist who has given the name. Yes, scientist who has given the name for mitochondria. Yes, TK Carl Benda. TK Carl Benda has given the name for mitochondria. Then what this Andrew Schimper did? We know symbiotic theory. TK symbiotic endosymbiotic theory for chloroplast. TK the chloroplast of plant. TK has been evolved due to endosymbiosis. This theory was given by Schimper. George Pallet, what he discovered? Ribosomes. Okay. Ribosomes, the name was given by George Pallet. Okay. The genes that express all the time and keeps up cellular process are called which genes? Which genes will be expressed at all the times? Whether it's an oncogene, protogene, or housekeeping gene or mutagenes. What will be the answer? Whether it's an oncogene or whether it's a protogene or housekeeping gene, yes. Housekeeping genes are expressed all the times. Okay, for example, we can say tubulin genes. Okay, these tubulin genes will be expressed all the times. Okay, for this is one of the Example for housekeeping gene. Okay. The country that deregulate golden rice recently to address the deficiency of vitamin A is which country? In 2021 they have done this one. Okay. Which country? India, Philippines, Indonesia, or Bangladesh? Yes, very good. Philippines. Okay. This is general agri question. Philippines. Okay. The type of protein that are involved in high order coiling of DNA post nucleosome stage are. They are asking specifically about post nucleosome stage. What will be the protein involved? Which type of proteins that are involved in coiling of DNA? Yes, very good, very good, very good. They are asking about post nucleosome. Okay, we know nucleosome will be there. It's an octamer of H2A, H2B, H3 and H4. Okay. At initial stage, the DNA will be wrapped around this. Okay, like this, it will wrap. But in post-nucleosome stage, non-histone proteins plays major role. Okay, for efficient coiling of the DNA around the nucleosome. Okay, so particularly they are asking about post-nucleosome stage. So non-histone proteins will come. Okay, clear? Another important question from electron transport chain. Okay, statement 1. Ubiquinone is a lipid soluble mobile electron carrier commonly referred to coenzyme Q. Okay. Second, ubiquinone acts as an electron sink which transfers electron from both NADH and FADH to next complex in electron transport chain. What will be the answer? Ubiquinone is a lipid soluble molecule. Yes, that we know. Lipid soluble as well as is a mobile electron carrier. Yes, that is true only. It will act as a sink, carrying the electron from both FADH and NADH. Then it will transfer the electron to complex 3. This also we have seen. So, yes, both the statements are correct. Very good. Both the statements are correct. Okay. If you want to practice topic wise objective question, kindly enroll in our test series. Okay. Those who are preparing for ICAR PG 2027, kindly enroll in our ICAR PG exam course. It will be highly useful for your preparation. Okay, students. First standard students will get discounts also. Okay. Polyploid with multiple complete genomes derived from two or different species is what? Polyploids with multiple complete genomes derived from two or more different species. They are saying from two or different more species. Yes, yes, very good. Alloploidy means from different species. Auto means from same species. Okay, from same species means auto. Then what is meant by aneuploidy? Aneuploidy means the change in the complete set of chromosome. For example, 2n minus 1, 2n plus 1, 2n minus 2. We have already seen this one. Monosomy, trisomy, nullisomy. These things are known as aneuploidy. Then what is meant by synthetic autoploidy? We will use here colchicine treatment for chromosome doubling. Okay, that is known as synthetic autoploids. Okay, clear? The process that operates in a diametrically opposite direction to CO2 fixation, leading to loss of photosynthetically fixed CO2. What is the process? In photosynthesis, CO2 will be fixed. There is another process where the fixed CO2 will be released. What is that process? Whether it's dark respiration or photorespiration, oxidative phosphorylation or decarboxylation. Yes, very good. It's photorespiration. Okay. We can also say a C2 cycle. In C2 cycle, whatever the sum of the CO2 fixed from photosynthesis will be released here. This will happen in chloroplast, peroxisome and mitochondria organelle. Particularly the CO2 will be released from mitochondria. Okay, clear? Primary role of post-transcriptional modification such as 5' capping and 3' poly A tailing of mRNA is what? What's the purpose? If you see the mRNA, there will be 5' cap and 3' poly A tail will be there. What is the role of this 5' capping and yes, very good. First role is stability only. Okay. To prevent this mRNA degradation by RNase enzyme, 5' capping will be there and poly A tail will be there. Okay. So stability is the right answer. Yes. Apart from that, during translation, this 5' site will act as this cap-like structure will act as a binding site for ribosome. Okay. Binding site for ribosomes. Okay. To initiate the translation process. Okay. Clear? Yes, clear. Inulin is an oligosaccharide of greater nutritional significance. Okay. It is formed from which of the following monosaccharide? I already told inulin is an polysaccharide. That too it is an homopolysaccharide. Homopolysaccharide of repeated fructose unit. This we have already discussed in our class itself. Okay. Fructone polymers will be there in inulin. Yes, very good. Okay. Which of the following rhizobial nod protein is activated by flavonoids, secreted by the roots of legume plants, and induce transcription of other nod factors after it activation? Yes. They are asking about here constitutively expressed. Okay. This is will target other nod genes for activation. What will be the answer? Other nod genes for activation. What will be the answer? Yes, very good, very good. Not D. Okay, not D. This I have already teached in nitrogen metabolism class. Okay. Not D is a constitutively expressed NOD factor that will activate the other NOD factors during nitrogen fixation. Okay. Then another most important thing you have to remember about this is these NOD factors are lipocyto oligosaccharide in nature. Okay. Lipocyto oligosaccharides in nature. This is also in important point. Okay, they are lipocyto oligosaccharide in nature molecules. Okay, this is also another important question. Okay, which of the following post-translational modification of proteins may tag them for degradation in 26S proteasome? What will be the answer? Which type of post-translation modification can tag them for degradation in 26S proteasome? Yes, very good. Polyubiquitination. This we have already discussed. 3 enzymes will be there. E1, E2, and E3. 3 enzymes will be there. Okay. These enzymes will add ubiquitin molecule to the target protein. If ubiquitin molecule was added with the target protein, that will be leads to degradation by 26S proteasome. Okay. This is clear for everyone. Okay. Then another most important thing I want to say here is this ubiquitin molecule. This ubiquitin, it is nothing. It's a protein. Okay. It is made up of 76 amino acid residue. Okay. 76 amino acid residue and its C terminal will have glycine residue. This is also another important question they have asked. Okay. 76 amino acid and its C terminal will have glycine residue. Okay. Another repeated question. Which of the following is a phosphorus containing membrane lipid? What is the example of phosphorus containing membrane lipid? Sphingomyelin, ceramide, sulfoquinosyl diacylglycerol, or cerebroside? Yes, very good. Sphingomyelin. Here, phosphocholine will be present as a head group. Okay, because of this, phosphorus will be present there. And another important thing you have to remember, SQDG, sulfoquinosyl diacylglycerol. Here it will have sulfate groups will be there. Okay. And if a plant is growing under phosphorus deficient condition, okay, if a plant is growing under phosphorus deficient condition, phospholipids okay, these phospholipids will be converted to sulfolipids. Okay, this is very important point. Okay, if a plant is growing under phosphorus deficient condition, okay, the phosphorus will be replaced by sulfate. Okay, that's why SQDG will be accumulated in phosphorus deficient soil condition. Okay. And nucleic acid molecule is a linear polymer in which of the monomer nucleotides are linked together by which bond? This will be linked together by which bond? Yes, hydrophobic interaction, ionic bond, phosphodiester bond, or hydrogen bond. What will be the answer? In nucleic acid molecule, yes, the nucleic acid molecule, I already told the nucleotides will be linked together by phosphodiester bond, PDE, phosphodiester bonds will be there. Yes, very good. Which of the following E. coli DNA polymerase possesses both 5' and 3' exonuclease activity and is thus have own proofreader and editor activity? Proofreading means what? 3' to 5' exonuclease activity will be there. Editor means 5' to 3' exonuclease activity will be there. What will be the answer? Yes, very good. Polymerase 1. Yes, very good. Polymerase 1 will have both exonuclease for 5' and 3' activity. Yes, very good. Which of the following is not a ketone body? Everything we have discussed in our lecture class itself. See, which of the following is not a ketone body? Acetone, acetoacetate, beta hydroxybutyrate, or isobutyryl CoA. Yes, very good. Isobutyryl CoA is not an ketone body. Okay. Ketone body biosynthesis will be happening where? Biosynthesis. This also I have, yeah, ketone body biosynthesis will happen in which organelle of which tissue? Which organelle of which tissue? Yes. Option A, mitochondria. Option B, cytoplasm. Option C, peroxisome. Option D, glycosome. What will be the answer? In which organelle the ketone bodies will be synthesized? Yes. Mitochondria of liver. Mitochondria of liver. Synthesis of ketone bodies will be happen. Okay. But it will be utilized. Ketone body will be utilized in extra hepatopic tissues. That means outside of liver only it will be utilized. Outside of liver only due to lack of the enzyme thiophorase. This is also another important point. Due to the lack of the enzyme thiophorase. This we have discussed in our class itself. Okay. These are important point to be remembered. Okay. The most abundant protein found in the biosphere. What is the most abundant protein found in the biosphere? Whether it's rubisco or peptase or pepcarboxylase or kinase. What will be the answer? Yes, very good. Rubisco. What is the most abundant carbohydrate? What is the most abundant carbohydrate then? Most abundant carbohydrate. Cellulose, chitin. What will be the answer? Most abundant. Inulins, fructans. Most abundant carbohydrate. Yes, very good. Cellulose. Okay, cellulose is the most abundant carbohydrate and most abundant protein is rubisco. Okay, clear? For thinning in apple, repeated question. For thinning in apples, the growth regulator used commercially is which growth regulator they will use for thinning of apple? Whether they will use NAA or 2,4-D, ethyl or GA3. What they will use for thinning? Yes, very good. For thinning, NAA will be used. Naphthalene acetic acid will be used for thinning. 2,4-D at high concentration, they can use as VD side. Okay. Ethyl, what it will do? It's a trade name for ethylene. Flowering in pineapple will be promoted. Okay. Then fruit ripening. These are the major thing. Okay. GA3 for internode elongation, alpha amylase activity. This will come. This is just for your revision. Okay. So for thinning, particularly they will use naphthalene acetic acid. Okay. Another important question from basics they are asking. Okay. Which of the following statement is true about D-glucose and D-talose? Okay. D-glucose and D-talose. What is the answer? Whether it's an enantiomer pair or whether it's an epimer or merely diastereomers and not epimers. D-glucose is aldolase while D-lactose is ketose. Yes, very good. Okay. You see here, if you see the structure of D-glucose and D-talose, this both are what? Both having aldehyde group at carbon 1. Okay. So, this both are aldoses only. So this option will be wrong. Correct. Second thing, if you see the arrangement of hydrogen and OH atom at carbon number 2 and carbon number 4, there is a changes are there. So it will not, it is not an epimer. Epimer means what? We at only single chiral carbon center, the arrangement of atom will be changed. So this also will not come. Whether this is an enantiomer, no, this both are D and D only. So this also will not come. So they both are mainly diastomers and not epimers. Yes, very good. Okay. Resistance developed by some weed against specific weedicides is due to, why it is happening? Resistance developed by some weeds against specific weedicide is due to whether the mutation of the genes will be happen or elevated expressivity of the gene, genetic recombination will happen or favorable environmental condition. This sure it will not come. Yes. If, for example, a particular gene is specific to that herbicide, if we mutate that one, that resistance can be happen. This we have already discussed in our plant biotechnology classes. Okay, you just go through this one. Okay, so gene mutation. Okay, a molecular marker that samples genome in a random and genome-wide manner is which marker? They are saying it's a random and genome-wide manner. Okay, the name itself it's there. Random and genome-wide manner, which marker? Yes, very good. RAPD markers. Okay, RAPD markers we can use. Okay, that can be used for genome in a random and in genome-wide manner. Yes, very good. RAPD is the right marker for selection this process. Okay, which of the following organelle contains enzyme that help in converting storage fatty acids into sugars in germinating seeds? We have discussed fatty acids are converted to sugars in which organelle? Which organelle? Which organelle? Oleosomes are what? Storage for fats. Storage organelle for oleosome. Yes. Glyoxysomes. Very good. Glyoxysome. Here glyoxylate cycle will happen. Glyoxylate cycle will happen. This will convert storage fatty acids into sugar. How it will happen? This is also important point. I already told. Okay. If fatty acids is there, that will be converted into acetyl-CoA. This acetyl-CoA will enter to glyoxysome. Okay. Here that will convert into succinate. This succinate will enter to TCA cycle. That will lead to formation of oxaloacetate. Okay. This oxaloacetate will be converted into PEP. This will be converted into glucose. Okay. By gluconeogenesis. Clear? So by this the fatty acid is converted into glucose molecules in germinating seeds. Yes, very good. Succinate, yes. So this is the process. So glyoxysome is the right answer. Okay. Again I am saying read the question twice. Okay. You will have sufficient time to answer the questions. Okay. Read question twice, grasp the question, then you go and write the answer. Okay. Don't click oleosome. Okay. Oleosome is a storage organelle. Okay. Oleosome is a storage organelle for fat. Okay. Clear? The plant cellular organelles that contribute to extra chromosomal inheritance. What will be the answer? Extra chromosomal inheritance will be there at nucleus. So sure it will not come. If they are already sold extra chromosomal inheritance. What will be the answer? Mitochondria, chloroplast and nucleus. Mitochondria and chloroplast. Yes, mitochondria and chloroplast is the right answer. Yes, they are semi-autonomous organelles. Okay. Semi-autonomous organelle. Okay. This is also important point you have to remember. Okay. In plant cell, non-pigmented plastids are called as what? What is the example of non-pigmented plastid? Amyloplast, yes, it's a storage plastid for starch. Chloroplast, it will be green in color. Leucoplast, colorless. Chromoplast, colored plastid. So what will be the answer? Non-pigmented means, yes, very good. Leucoplast. Leucoplast. An example of crop where the reserve food material in seed is stored in endosperm. This they have asked in 2021 also. Endosperm speed is what? Castor, yes, very good. Castor, we have already discussed yesterday only. Castor will have endosperm storage things. Okay. Identify the specific DNA sequence by southern blotting involving the following procedure. They have given the different steps for southern blotting. Okay. 4 different steps they have given. We have to arrange that. What are the steps they have given? Digestion of the DNA with the restriction enzyme, fractionation of DNA fragments, transfer of single-stranded DNA molecule from gel to an absorbent sheet, and the fourth statement is hybridization of radioactively labeled probe to target DNA. What will be the correct sequence? What will be the correct sequence? Yes, ultimately we will do hybridization at last only. Okay, that is correct. First we will digest the DNA with restriction enzyme. Yes. After that what we will do? We will run it. That means we will resolve on the agarose gel. This also correct. A after B will come. Yeah, A, B, C, D. Yes, very good. This will be the right option. The natural mode of exchange of genetic material in bacteria. Natural mode of exchange. They are asking particularly natural mode of exchange of genetic material in bacteria. What will be the answer? Natural mode. Transformation is different. Transformation we will do in lab conditions. Transduction means what? That is also different. Bacteriophages will be there. Transfection means we will use some forces. Conjugation, yes, conjugation. Here F plasmid will be there. Okay, F plasmid will be there, or we can say what we can say. Here, sex pilus will be there. Through sex pilus, the genetic material will be transferred. Okay, yes. So, the right option is conjugation. Another important question, okay, this you have to observe properly. Okay, another important question. Okay, a protein that has 8 cysteine residue. What they have given? Protein has 8 cysteine residue. How many possible ways are there to make 4 disulfide bridge in its structure? Okay, they are given, yes, they have given 8 cysteine residue are there. What are the possible ways to make the disulfide bridges? Okay, you see 1, 2, 3, 4, 5, 6, 7, 8. Okay, 8 things are there. Just you imagine this has 7 different possibilities. This can bind with this also. Okay, this can bind with like this. 7 different possibilities will be there. This one can bind with 2, 3, 4, 5, 6, 7. So 7 different possibilities will be there. Okay, if you see this one, the second cysteine residue, this having 6, 1, 2, 3, 4, 5, 6. 6 different possibilities base will be there. If you see the third cysteine residue, it will have 5 different possibilities, and this will have 4, this will have 3. At last, this both will be involved in disulfide bond formation. If you multiply all these things, it will come 105. I hope this is clear. Just you have to like apply the things. Okay. So 8 cysteine residues are there, but 4 different types of disulfide bond will form. Means it's just a probability how it will go and bind. Okay. So the right answer is 105. Okay. The bioinformatic tool used to identify microsatellite repeated motif in a genome is what? Microsatellite repeated motif in a genome. How they will do? Whether primer 3 plus? No, this is used for primer designing. This will not come. MISA, yes. The name itself is there. TK, microsatellite tool. TK, yes, microsatellite tool. So this will be there. SNP digger, it will not come. Codon code aligner also will not come. TK. So bioinformatics, sure they are asking 2 questions. TK. Bioinformatic tool, sure they are asking minimum 2 questions. You have to be read properly. TK. Corresponding a genetic disorder by the way of introduction of normal gene in place of missing or defective one is referred as. Definition. What is gene therapy, gene conversion, gene donation or regulation? Yes, very good. Gene therapy. Okay. If a particular thing is defective, we will do gene therapy to replace that one. Okay. So Gene therapy will be the right answer. The instrument used for measurement of water potential of plant. Water potential of plant will be measured by which instrument? Water potential of plant will be measured by which instrument? Whether hygrometer, no, this will for measuring the moisture, air moisture we can measure. Porometer, stomatal conductance we will measure, this also will not come. Yes, pressure bomb apparatus, yes, pressure bomb apparatus. IRGA where we will use? This for photosynthesis measurement, IRGA we will use. So, pressure bomb apparatus will be the right answer. Then another most important thing I want to say, if we want to measure the water potential of plant, we will use pressure bomb apparatus. If we want to measure the water potential of soil, what we will use? General agree. If you want to measure the water potential of soil, which instrument we will use commonly? Which instrument we will use? Yes, I need the answer from you. Which instrument? Lysimeter, tensiometer, these things they will use. Okay, clear? So you have to read the question properly, whether they are asking in plant or whether they are asking in soil. Okay. Next, the water potential of pure water. This they have already discussed only. What will be the water potential of pure water? It will be -1 or 0 or +1 or infinite megapascal. What will be the answer? It will be -1. Yes, very good. Water potential of pure water is 0. That is the maximum water potential. Okay. That will be the maximum water potential. Okay. The activity of enzyme pyruvate dehydrogenase is regulated by which of the following modifications? The activity of enzyme pyruvate dehydrogenase, that is PDH complex, is regulated by which type of modification? Whether carboxylation, methylation, glycosylation, or phosphorylation and dephosphorylation. Yes, phosphorylation and dephosphorylation. If this PDH complex is phosphorylated, it will become inactive. Okay. If it is dephosphorylated, it will become active. This we have already discussed. Okay. This we have already discussed in our metabolism class. Okay. The crop that flower when the day length exceed a critical duration in a 24 hours. Okay. For example, this is the day length. This is equal point. If the day length is exceeding, okay, more day length is there, means whether it's a short day plant or long day plant or day neutral or short long day plant. Yes, very good. It's a long day plant. Okay, it's a long day plant. Very good, long day plant. Okay. Nobel Prize was awarded for fire and mellow for the discovery of, for what purpose? For discovery of what purpose? Okay, fire and mellow awarded Nobel Prize for discovery of CRISPR-Cas technology. No, CRISPR-Cas technology was not given by them. Yes, RNA interference. Okay, RNA interference in C. elegans. Candidatus elegans. They have done first illustrated this. Yes, 2009. Very good. 2009 Nobel Prize. Okay, fire and mellow. Okay, RNA interference. Another simple question. Cryopreservation of plant material using liquid nitrogen. This temperature is important, -196 degrees Celsius. For conservation of eventuality is here. Whether it's a short-term process or long-term strategy or headache strategy or medium-term strategy. Yes, very good. It's a long-term storage or preservation of the materials will be done by using cryopreservation, by storing the samples in liquid nitrogen. The temperature is most important. -196 degrees Celsius we will use there. Okay. The nucleotides differ from nucleosides. Nucleotides is differ from nucleosides with respect to whether the sugar backbones will be changed or phosphate backbone will be differ or nitrogen bases will be differ or sugar and nitrogen base will be differ. What will be the answer? Yes, very good. Phosphate. Nucleotide means sugar plus phosphate plus base. Nucleoside means sugar plus base. I hope this is clear. This they have asked in 2019 also. Okay. With this, we are completing the first session. Okay. I will see you all at 2:30. Okay. We will complete the PYQs for 2020 in afternoon session. Okay, students. If you have any doubt, kindly drop the message here. I will reply you. Okay. Thank you, students.